folks. So it's my pleasure to introduce my colleague, Professor Giovanna Ghirlanda, and we were hoping to have her here with us in person, but Mother Nature had other ideas. So luckily, COVID prepared us for such occasions, and so here we are back on Zoom. Giovanna is a professor in the School of Molecular Sciences at Arizona State University. Actually, we first met when I think I was finishing up my PhD, and you were just starting a new professor there, a new assistant professor there. Wasn't as long ago as you all may think. Anyway, she earned her PhD from the University of Padova in Italy, and working on supramolecular chemistry of metal complexes in a lot of membrane materials, micelles, and liposomes. And then went on to do a postdoc with Bill DeGrotto back when he was still at UPenn. I think he's over at UCSF now, I think. Is that right?
He is correct. That's exactly right.
Yeah, and worked with Bill on de novo design of multimeric proteins and heme binding proteins. And recently your group's been focusing on designing these protein-based hybrid metalloenzymes that catalyze light-driven hydrogen production and carbon dioxide reduction. And that's what she's going to be talking to us today about metalloprotein engineering in applications for renewable energy. So with that, I will hand it over to you, Giovanna, and thanks again. We're going to have you here again. As I mentioned, we're going to follow up with this and bring you in person because really want you to meet with our students, and there's a lot of faculty that are really looking forward to engaging with you as well.
Okay, so thank you for the kind of introduction. And yes, absolutely, I would love to come up and see all the wonderful work you guys are doing in NEU. I mean, we learned a little bit last year when Gabe and I were involved with others in a proposal, but I've been wanting to circle back ever since. So as Gabe was saying, yes, we're working on engineering metalloproteins, and we are particularly interested in application in renewable energy. And so I probably do not need to convince you that, let me see if that climate change is becoming obvious at this point throughout the world. And, I would argue that the signs of climate change, of global warming, have been evident for a longer time in Europe than they have in the US, which kind of explains a little bit of the difference in approach, in terms of policies and regulations in Europe and in the US. But this is a picture of a glacier, the Marmolada Glacier in Italy, in the Italian Alps. And this summer in July, basically the whole, half the glacier collapsed, so you can see this rock that kind of detached. And unfortunately, between 10 and 12 climbers lost their lives on that. And the reason this was, kind of brought it home for me is because I did this hike when I was in graduate school, and it's basically a hut to hut across the glacier, and it's obviously beautiful. But seeing this glacier disappearing, and that's the fate that many glaciers are encountering now in the Alps, is really highlighting the effect and the tipping point that we are going towards in terms of climate change. And there is a little doubt that this is climate change effects that we are seeing are linked to CO2 emissions and increase in CO2 emissions linked to human activities, and particularly as we can kind of follow the carbon dioxide emission coming from coal, oil, and gas as tracking with the CO2 emission and then changes in temperature that we observe in the global level. So what can we do to address this issue? So what my group got involved in this work basically several years ago when we were part of a DOE EFLC grant based here at the ASU. But the idea is to utilize solar light as a source of energy to power several transformations. And so the transformations we are interested in are both the production of fuel, so directly something that can be utilized to power the economy, and they can be either carbon neutral or carbon free, but also utilizing solar light to drive the production of energy-rich chemicals, or also to make, for example, intermediates in synthesis for pharmaceutical industry. And the idea is to utilize these resources like water, carbon dioxide, nitrogen to make this more elaborate molecules. And so the idea is, pretty much basically summarized here. I should say here, where we have basically an artificial enzyme that now interacts with a cofactor, and upon excitation of, by a photosensitizer, in turn excited by light, forms hydrogen. So let's look a little bit more at the kind of reactions we are interested in. So we are interested in hydrogen production. We are interested in reducing CO2 to carbon monoxide, and hydrogen to make syngas, but also to make possibly methane, and even to make carbon-carbon bonds. And the other reaction that is really important in terms of energy neutrality is the reduction of nitrogen to ammonia. And this nitrogen fixation controls basically the agricultural production worldwide. And so it's very important to be able to do this in a sustainable manner. So in nature, the enzymes that catalyze this metabolism reaction, in which basically gas gets utilized or produced, are catalyzed by enzymes such as the hydrogenases, carbon monoxide dehydrogenases, and nitrogenase. And they have one thing in common. They are all containing iron sulfur clusters in their active sites. And this points to a very ancient origin for those enzymes. And in fact, they were evolved in a pre-photosynthetic world where basically the atmosphere on Earth was highly reducing. And so there was no oxygen around. And then, of course, the advent of photosynthesis completely changed the face of Earth, and basically resulted in changing also of the content of oxygen and of the redox potential in general of the environment. And so, interestingly enough, basically these enzymes are still oxygen sensitive. However, organisms that utilize them have come up with methods to both make them and also keep them anaerobic during their activities. And we'll talk a little bit more about that. So the enzyme that we have used, we looked at first, and kind of a prototype for this kind of enzymes is the hydrogenase. So this is the easiest redox reaction you can think of, basically taking 2 protons, adding 2 electrons, boom, making hydrogen. And this can be done reversibly. In fact, basically microorganisms that make hydrogenase use it in both directions. They produce hydrogen, and so they use proton as a acceptor of electrons, basically when in metabolic conditions in which there is an excess of electrons that needs to be disposed of. But also we have microorganisms that utilize hydrogen present, for example, in hydrothermal vents at the bottom of oceans, to utilize hydrogen again as a source of energy. So they extract electrons directly from hydrogen. And the enzyme has been very well studied, especially electrochemically, using a technique called protein film voltammetry, in which proteins are absorbed on the surface of the electrode. And that allows investigators to basically interrogate very specifically the redox potential of all the centers, but also the mechanism of action. And one can probe it further by changing the environment in the solution. And it turns out that this is really a bit of luck that this enzyme was picked, and of course was picked because of its interest in the context of making hydrogen in a sustainable manner. But they were so lucky because these enzymes contains an electron conduit made by iron sulfur clusters that basically brings electrons to the active center. And literally because of the electrostatic charges rather on the surface of the protein, when put on graphite electrodes, the protein literally sits with its butt basically on the electrode, and allows for very easy communication with the electrode. And so that was a lucky thing, meaning that many other redox proteins are not so easy to study with this technique because of their on how they absorb on electrodes and also the heterogeneity of the deposition process. But going back to us, this enzyme is made by a ferredoxin-like domain. So it's basically a tiny little protein, which in fact in some species is a separate protein that binds two iron sulfur clusters and basically is simply in charge of transfer electrons to and forth. And the active site is made by another iron sulfur cluster, what we call the cubain type. They're called cubain because you can see them here. They're literally little cubes, and they're made by 4 atoms of sulfur and 4 atoms of iron. And incidentally, those form spontaneously in hydrothermal vents, and they're stabilized by small thiolates, such as thioethanol, for example. And so the active site contains one of these cubain type, basically linked through a cysteine to this other iron sulfur cluster, now a 2 iron to sulfur cluster, that is weird from an enzyme point of view because it contains no amino acids. So the two metals are coordinated exclusively by a small gas ligand, so CO and CN, and also by this kind of bridging ligand that contains a nitrogen. And so the theories on evolution of these enzymes point to the fact that probably the first hydrogenases evolved to take up from the environment something that was already present. And recent studies by David Britt and others have now elucidated that extant organisms make this active center by catabolizing tyrosine, which is the source of the CO ligand and the cyan ligand, and serine to make this ligand here, right? Cysteine, rather. So, I mean, from an energetic point of view and metabolic point of view, this is completely wasteful because the microorganism goes all the way to making an amino acid and then takes it apart to make a small 2-atom molecule. And it really points to a phenomenon that we see often in evolution where basically the enzyme is evolved in center condition and then the organism gets stuck with that solution in a local minimum and cannot basically rewire to make a completely different hydrogenase, but rather, evolves ways to make and come up with those small pieces that are no longer in the environment. So going back to us, I trained as an organic synthetic chemist in a previous life. And so the way I think about this kind of problems is retrosynthetic. So, my group looked at the whole enzyme and then divided it up in a ferredoxin-like domain and a catalytic domain. And then we basically tried to, divide and conquer and address those two problems separately, which is also very helpful in terms of not burning too many graduate students in the process. So how do we do that? Let's start with looking at the electron transfer process first. So what we wanted to do is to be able to incorporate iron sulfur clusters wherever we wanted in the protein, basically at whichever distance and environment we wanted. So we started by taking from the PDB, so then the repository for protein structures, the iron sulfur cluster of this particular organism, and we picked it for a number of reasons that we can go into. But basically, the main reason is that it now has 2 amino acids here that are in a helix arrangement already, and the other 2 that are distal in the sequence. However, they are close enough in space that can be kind of recapitulated again on a helical structure. So we looked at all the requirements in terms of geometry and sterics, and so the distance between sulfurs and so on and so forth. And we realized that we're indeed compatible with the helical structures. And so what we did was taking a protein that we that had been designed basically many years ago. In fact, I did this as a postdoc many months ago. This is a de novo design alpha helical protein. It's a dimer of two basically peptides, then one can synthesize very easily. And the way it works is that, we have two long helices pairing against each other, and then one short helix per dimer basically kind of docking against this pre-existing structure. And this turns out to be an absolutely super stable peptide, and so it's very good starting point in terms of making mutants. So what we did basically using PyMO, a protein visualization software, we just overlaid the iron sulfur cluster of Saratoga maritima, the one that we just described, with the cluster with the cysteines surrounding it, and we kind of overlaid it in the structure. One nice thing about de novo design proteins, and particularly these helical ones, is that they're built on a helical repeat. And so the sequence is described by 7 residues. So, typical hydrophobic, hydrophilic, hydrophilic, hydrophobic, and then hydrophilic, and so on and so forth. And basically, this makes the sequence very modular. So once we figure out how to put a cluster in one position of the coil, we can easily move it up and down the coil. So we made this protein, and we had basically designed it so that distance between the two clusters will be about 35 angstroms. We made a cluster. We did the EPR, basically showing that the reduced species has exactly the signatures that one would expect by EPR. And also we used this Eldor experiment to kind of simulate and calculate the distance between the cluster, which turned out to be 36 angstroms versus the 35 in the design. So, we're doing pretty good for that. And then, basically several students in my group played several games, such as moving the clusters closer together. And as I said, this is easy to do because of the way that the sequencing coil works. And we now have something in which the clusters are remarkably closer than they were before. And once again, this design has the EPR signature that one would expect. Also has a redox potential of minus about 0.5 volts. And then again is consistent or similar to what observed in natural ferredoxins. And this was quite an achievement when we got to this, because it's basically other groups have failed to reproduce the electrochemical signature of iron sulfur cluster proteins, and which, because the cluster will decompose during the redox change. And so what we did with this protein now is showing two things. First, that we can photoexcite a zinc porphyrin and use it to reduce the cluster. And we can use transient absorption kinetics to show basically how the protein quenches the photoexcitation of the zinc porphyrin. We can also use the reduced species to reduce cytochrome C, which is basically a natural protein, and basically making this primordial electron transfer chain between two proteins. We also played with other structure, and so we used repeat proteins in which we have a helix loop helix motif that repeats, a number of times, and one can use it to make basically very long fibrils. And we collaborated with a group in Spain for this work because they had experience using these proteins as biomaterials. And we use our de novo design tricks to insert one of these cubane type clusters in the sequence. And we made basically proteins with 1, 2, or 4 clusters. And we showed, first of all, that the proteins are remarkably stable. And we also showed that, again, the clusters have the signature one would expect for natural iron sulfur cluster proteins. And also the redox potential is in the range that we'd like to see. And interestingly, basically, the clusters are not coupled, so they're actually not talking to each other. And the redox potential is very similar. However, this difference between oxidation and reduction wave kind of increases with the size of the protein. We also showed that we can observe basically photoexcite the cluster and observe basically the communication with aromatic residues in the protein nearby, kind of quench basically the cluster excitation. And so with that, now what we want to do is make this more complex protein structures where we can basically, for example, put a mimic of the antenna system on one end and then use this chain to transfer electrons to, for example, materials or to catalytic proteins. So with that, the second aspect that we focused on was making the catalytic domain. In other words, make now an artificial enzyme that will work as a hydrogenase. And if you remember, we talked about how this active site contains no amino acids. So we couldn't quite use the same trick that we used before, but we had to figure out a different approach. And we basically built on work by organometallic chemists who have gone nuts over the years in trying to reproduce the spectroscopic and activity profile of natural hydrogenases by making simplified models. And so what we ended up doing is thinking, well, instead of trying to bind, the irons with amino acids, what we can do is design an artificial amino acid in which now the bridging ligand would be the side chain of an amino acid. And so we made these two artificial amino acids, and then we kind of worked to see if we could introduce them in a peptide to make a hydrogenase. So we did exactly that. We used a very simple helical peptide, and my student in India basically synthesized it on a peptide synthesizer with this artificial amino acid, and then reconstructed the diiron cluster onto this amino acid. And we showed basically that we have incorporation of the cluster, just like one would expect. The UVV signature and the FTIR signature are similar to what is seen basically in hydrogenases. And most importantly, we can now observe some pathetic reduction of proteins into hydrogen. So the turnover numbers are very small, but this was published now 10 years ago, but was the first demonstration that we could actually make hydrogen with an artificial hydrogenase. And the way we do this is, we photoexcite ruthenium bipyridine. In terms, this will reduce the active site, and the active site will catalyze the reduction of protons to hydrogen. And then we use the sacrificial electron donors such as ascorbate to repristinate the ruthenium. So what you see is that we have turnover numbers of about 80 in the presence of light. We can also immobilize these peptides on electrodes, and then we observe basically this reductive catalytic wave corresponding basically to reduction of proton to hydrogen. And not surprisingly, the size of the wave, so the amount of current or the amount of reaction that we have, is very strongly dependent on pH. And basically the breaking point is between 5.5 and between 4.5 and 4, and that corresponds to the pKa basically of the group. Now, that was not quite a protein, so we have also inserted that helical protein within a more complex protein structure, and we show that the protein is now decently well folded. It's also quite stable, and now the turnover numbers increase by a factor of about 3. And most importantly, we now have a catalyst that sustains the activity for more than 15 hours. So we have something that is actually remarkably stable in terms of activity over time. We have also used another approach, basically copying an approach published by George Whiteside since 1978, which is a very long time ago. But Whiteside had this great idea of exploiting the non-covalent recognition between biotin and streptavidin. You most likely have heard about this. because it's basically the strongest interaction known in nature. The PK, and I mean the KD for binding of biotin to streptavidin is about 10 to the minus 14. It's insanely strong. And so Whitesides had this cute idea of taking biotin, modifying it with an organometallic center, in his case, a rhodium complex, and then observing the activity. And the activity he was looking at was an antiselective reduction of a double bond to an alcohol. But, despite that, what you observe is a remarkable increase in antiselectivity, because now the complex is in a protein environment, so it's chiral and cannot basically that forces a stereoselectivity. Now, in our case, obviously, reduction of proton to hydrogen is not so stereoselective, but we were interested in the concept of protecting the active site from degradation. And so it turns out that, by doing this, the system works perfectly well for us too. And we got the crystal structure of the complex within streptavidin. Unfortunately, in the crystal lattice, the two proteins can kind of abut to each other. So we kind of lose electron density near where they touch, because probably there is some floppiness there. Nevertheless, that proved to be a valid approach. And in fact, we can see from the sharpening and narrowing of the CO bands in the FTIR spectrum, if we compare the complex in solution with the complex in the protein, then the narrows get, the bands get more narrow. In one case, split in two, and that basically indicates that the complex is now not free to move around as much. And most interestingly, obviously, we have also an increase in catalytic activity. So here we are comparing basically, streptavidin is just our control, but then we are basically comparing our artificial hydrogenase, which call strep hydrogenase. And we are observing the same concept where, at lower pH, we have an increase in activity. We can also see an increase in activity compared to the isolated biotinylated complex when the complex itself is in the protein. And we have an increase of about 10-fold. Most importantly, we were able to link the increase in activity to an increased lifetime of the catalytically active species. So again, what happens in these reactions that we use light to excite ruthenium by pyridine to the photoexcited compound, that in turn reduces the complex, and the active species is thought to be formally an iron 0, iron 1, and that is what carries out the activity. And then again, we use ascorbate on the back end to reduce the complex. So the species that is active is the blue species. In the complex itself, the lifetime of the blue species is basically about 490 microseconds. When the same complex is in streptavidin, its lifetime is basically increased by a factor of about 4 or 5 to 2.4 milliseconds. Well, I'm not going to go over this, but basically we collaborated with a group in Spain to use the same concept to introduce different kind of cobalt complexes into streptavidin. And we did a lot of mechanistic studies to see what explained the increased activity in streptavidin compared to the solution phase. And turns out to be basically, in this case, what we think are specific interactions with some groups in the streptavidin molecule. So, with this, we can indeed incorporate hydrogenase mimics into the scaffold to make artificial hydrogenases. However, the problem is that the catalytic activity itself is pretty, it's not ideal. And so one of the things we were interested in doing too is to ask the question, what would a hydrogenase look like if it were to be evolved now in photosynthetic world where the atmosphere is highly oxidant. And clearly, it would not look like what we have so far, because we would need something that can be oxygen tolerant. So we kind of looked at the literature, and turns out that cobalt porphyrins can also reduce protons to hydrogen. And here there are two types that were published now a while ago. But one type in particular was interesting, and this came from the Nocera group. What they showed here is that when one introduces a carboxylate near the active site, now the catalytic efficiency of the cobalt porphyrin towards hydrogen production increases. And so the hypothesis they were making is that, basically this group will act as a shuttle for protons. I should say that, in this kind of reactions, what happens is that a proton coordinates to the cobalt, gets reduced to hydride, and then the hydride picks up a proton to form the molecular hydrogen. Now, all these porphyrins were not soluble in water, so they could not be used in the conditions we are interested in. Another interesting aspect of cobalt porphyrin chemistry is that they can also reduce CO2 to CO and to formate. So we were not the only ones who have the same idea. In fact, there's exactly the same time, a group in Rochester, Cara Brandt's group, came up with exactly the same idea using a basically a fragment of cytochrome C. They were using cobalt cytochrome C. And they also showed basically, in their case, electrochemically, hydrogen production. But then one of my students did the same thing, and we used two scaffold. So we used basically a cytochrome B562, and we also used myoglobin. Why did we make this choice? Well, we wanted now to look at proteins, natural heme-binding proteins, for which protein-protein interactions are already existing in nature. And so we were thinking of potentially utilizing these modules as electrolyte modules, both in vitro and in vivo. And so my students over the years made a bunch of mutants to this protein, and just quickly we are going to look at one of them in particular, the cytochrome B562 scaffold. And so the natural protein coordinates heme and coordinates it axially through a methionine and a histidine. And what we decided to do was to change the identity of these amino acids, and also to change the identity of the metal from iron to cobalt to basically impart to it the hydrogenase activity. So we showed that indeed these proteins bind cobalt porphyrin, and so they have no problem accepting a change in metal. And the KDs are in the nanomolar range. We also showed that binding to the cofactor increases the stability. So here in black is the apo form, and when we add the porphyrin, we have an increase in stability. At least when we are looking at methionine substituents, when we are looking at basically at histidine substituents, not so much. And the reason for it is quite simple. If you go back to the structure, the histidine here is at the end of the protein on a helical structure. So basically binding, coordination by the histidine to the metal results in folding of this little piece of helix. And if we change the histidine to anything else, now the protein doesn't fold as well, and so remains partially unfolded. And that's exactly what we are observing here. Now, going back to us, we looked at all, again, several mutations. And initially, the hypothesis we were making is that by adding a carboxylate, so either aspartate, so D, or glutamate, and that's E, to the axial coordination, we would increase the ability of the protein to act as a proton shuffle, and so increase activity. And turns out it didn't work that way. So here in black, we have wild type, the cytochrome, so with methionine histidine ligation. When we change methionine to glutamate, we have an increase in activity and a further increase when we add aspartate. And because of the length of the side chain in these two groups, it actually made sense to us that aspartate will be exactly above the cobalt. But then, of course, we also did the control where we take out that methionine and we put an alanine instead. And alanine is a residue that doesn't have functional group on the side chain and so cannot coordinate to the metal. And what we observed is that, actually activity increases even more, probably because we are now forming a cavity on top of the cobalt and that can accommodate water. So with that, basically, we show that, these proteins indeed can serve to produce hydrogen. And they can do that in a completely different way than hydrogenase does. And of course, the turnover numbers are nowhere close to what the enzyme has, but nevertheless, we can modulate it by changing the redox potential of the protein and by creating a cavity. Most importantly, what we can do is now carry out the reaction for a long time, and so over 15 hours, in a photoinduced manner, which the hydrogenase cannot do. And we can do it at increasing concentration of oxygen. And so we do take a hit in the activity, but if we compare the reaction run in anaerobic conditions here in black, with the one run basically in open air, so with almost 20% of oxygen, we see that, we still have about 1/3 of the activity, whereas hydrogenases would be completely dead in those circumstances. The other thing we did was to look at carbon dioxide reduction. And so we showed that indeed in water, we can form two things. When we expose the protein to a saturated concentration of carbon dioxide, we also reduce it to carbon monoxide and to formate, which is not in the slide because we have to, while we can detect hydrogen and carbon monoxide simultaneously using gas chromatography, we can only observe formate kind of post-mortem, so as an endpoint, because we observe it by NMR. So what we should, going back to our mutations, is that while we have a remarkable modularity and modulation also of activity, in terms of hydrogen production here in blue, the CO2 reduction is minor and it's also less sensitive to mutation. All the mutants, however, are more active than the isolated cobalt porphyrin. So with that, let me switch in gears a little bit. And so one thing that we are very interested in doing in my group is going now to in vivo conditions. In other words, utilize these synthetic proteins as part of either of synthetic cells or of natural cells. And one thing we were particularly interested in doing is to generate basically organelles that can work to reduce CO2. And the inspiration for that is a carboxysome, which is an organelle found in photosynthetic organisms that serves to basically increase the activity of rubisco. Rubisco in nature is an enzyme that fixates CO2 by basically starting with a substrate and adding an atom of carbon to it. And this is part of basically metabolism in cell. So what a carboxysome does is to form a shell made by proteins, which contains both rubisco and also basically carbonic anhydrase. And what that does is to take bicarbonate and basically release back CO2 in situ. So by having these two enzymes co-localized, we can increase the local concentration of CO2 and also, protects the rubisco from oxygen damage. So we were wondering if we can, kind of utilize artificial protein now to make something like that, right? And so one thing that we wanted to do is to avoid basically utilizing protein shells, because we wanted to keep the system as simple as possible. And so basically in cells, basically eukaryotic cells are basically surrounded by a lipid bilayer, but also contain separate internal organelles. And those organelles can in turn be surrounded by a liquid bilayer. So for example, the nucleus is, and that serves both as a barrier and serves to compartmentalize basically certain reactions. But the biggest discovery in biology, arguably in the last 10 years, has been the fact that cells also contain membraneless organelles, such as nucleoli and the causal body, which are not surrounded by bilayers, but they look like basically droplets in the cytoplasm. And for the longest time, people thought that those droplets were basically just protein aggregation and were absolutely of no biological relevance. And turns out that instead they are absolutely fundamental in regulating, for example, protein translation, and also they're involved in viral infections, and so the way by which viruses insert and take over hosts. How does this work? Well, it's really based on the physics phenomenon, and so it's a phase separation of a liquid phase within another liquid phase, where basically the light blue is now the cytoplasm and the dark blue is our proteins of interest that are coalescing or conservating into a liquid form. And pH, temperature, concentration, binding patterns, and other factors contribute to switching this equilibrium. So we can literally define phase diagrams in which we have proteins that are soluble in the cytoplasm here on the outside, and protein that coacervated within the cytoplasm here in the inside. Generally speaking, these processes are mediated by unfolded or disordered proteins, because we need the flexibility for forming these compounds. And so what the group is working on right now is to form this membraneless carboxysome. And we are going to do that, or we have done it, hopefully, by complex coacervation. So in our case, we are taking a protein that has a large negative charge, and that's our cytochrome by B562. We now re-engineer its outside surface to be negatively charged. And then we're going to mix it with polyarginine, which is basically polycation polymer, in order to achieve this complex coacervation in which the protein will now fold and form these separate droplets, then can in turn accumulate ruthenium, piperidine, and other co-reactants. So why did we choose supercharged proteins? Well, the idea, again, was to mimic nature a little bit. And so in nature, we have mRNA, which is, polyanion polymer, coacervating with specific proteins that bind to it. And those tend to be cationic, so positively charged. And so that complex coacervation with attraction between a negatively charged polymer and a positively charged polymer basically supports the formation of the membraneless organelle. So we thought we could do the same thing with the complication that when we supercharge our protein, the protein is going to be unfolded. However, we can rescue partially that folding, by adding salt or polyarginine. And then in turns, when we add the ligand, so cobalt protoporphyrin 9, we can basically kick a little bit more the protein towards folding, and we can obtain a structure that now resembles the native structure. And this work was done in collaboration with a friend of mine, Ron Coder at CUNY published it on PNAS a few years ago. And he basically was studying this phenomenon from the point of view of folding. So we use the same concept, and so we supercharge our cytochrome, and then we showed again that we can fold it either with salt, or we can take basically this point here where the protein is still unfolded, add the porphyrin, and obtain a completely folded protein. So we can recover a folding in those conditions. The other thing that we've shown is that when we mix our supercharged protein with polylysin and with porphyrin, we can obtain this phase-separated droplets. And so this is a confocal microscopy, basically showing dark side and bright side, and then the overlay, and basically the overlay. And so we can see that basically the droplets are indeed fluorescent red because of the porphyrin that they contain. And so all the protein appears to be successfully within the droplets. And the other thing we can do is use basically CD spectroscopy to show that the protein in the droplet here in green is folded just like we would expect, and very similarly to wild type. So specifically what we have here is the basically the protein by itself is completely unfolded. Polyarginine is also completely unfolded here in black. When we mix the two and we have these droplets, we observe, the blue spectrum, and by subtracting polyarginine, which is unfolded, we get our helical structure. We also show that the position of the Sorey band in the UVs in these coacervates is at basically 427, which is exactly what we would expect for a cobalt porphyrin that is coordinated between the cytochrome. So with that, we show that, we can make these droplets, they will contain our protein. And the last thing to show now is whether or not these droplets are catalytically active. And so we tested both hydrogen production and CO2 reduction, and basically at the same time. And so what I'm comparing here is turnovers observed in photocatalytic conditions for free cobalt porphyrin, for the cobalt cytochrome folded with salt, and for the cobalt cytochrome folded, and in droplets in the presence of polyarginine. For hydrogen, it's very clear. We have increasing activity from the free porphyrin to the one in the protein to the protein in coacervate, which is the most active of the series. For CO2 reduction, we still have an increase between solution and cytochrome, but we don't have much of a variation whether the protein is in coacervates or in solution. And we're still kind of, trying to clean up this data and investigate exactly what's going on. Part of the problem is that, when we have coacervates, the solution is turbid, so light penetration is not as efficient as it is in solution. So with that, I just wanted to basically do a quick recap of what we discussed today. So basically, my group is interested in expanding the protein function, so designing an artificial protein that can have different activities than those observed in nature. We oftentimes do it by interfacing an organometallic site with a protein, so generating artificial enzymes that don't look like the ones in nature. And so we've done work to control electron transfer. We've done work then to design redox pathways in which our designed proteins will interface with natural proteins. We have also shown that we can incorporate two different types of artificial active sites within proteins. We are now working on re-evolving these proteins around artificial active sites, mimicking basically the evolution of enzymes. And we also shown that we can increase the complexity of the system even further, and going towards synthetic biology by designing artificial organelles that contain proteins of interest and basically can regulate activity. And so we are playing a bunch of games to basically link their activity that we are interested in with the formation of the droplets. And, I didn't have time to go too much into that, but basically the droplets indeed suck up also the ruthenium bipyridine photosynthesizer, and that's probably one of the reasons for the increase in activity. And so the idea is by modulating formation of the organelles, we can also modulate activity. So with that, I just wanted to basically thank my students. Most of the work was actually started by an amazing graduate student, India Roy, who's now a senior scientist in David Baker's lab at University of Washington. And he's starting his own company now, so doing fantastic work there in translating his research to products that are applicable. And then the work was continued by Rafael Alcalá-Torano, who's now at Tufts as a postdoc. And currently in the lab, we have Michelle and Abesh. Basically, they started, Michelle especially started the whole work on liquid-liquid phase separation. Nick Halloran is now a scientist in a startup company in San Diego. And over the years, we have collaborated with several people, and particularly for the hydrogen catalysis with Julio Loré-Fillol in Spain, in Tarragona. And with that, basically, thank you, and I'll be happy to get any questions.
Thanks, Giovanna. Yeah, I want to open this up to questions, and folks, you can either, I would just, you could raise your hand or just unmute your mic and speak up.
I do have a question. So in one of these slides, you spoke about interfacing with photosynthesizers.
Yes.
Natural photosystems and multifunctionality in proteins. Could you please just explain this a little?
So I guess we're talking about here. Let me get down to and see if that's what we are all talking about. You talk about this?
Yes.
So, okay. So the idea is to, in the case of these repeat proteins, as I said, my collaborator, Aitziber Cortajarena, who's in San Sebastian in Spain, utilizes this protein as components for biomaterials. And, because they form this kind of a super helical coil and, fibrous structures that can be decorated with, several things. So, for example, they worked in inserting gold nanoparticles on top of this. The advantage here is that we have a protein that is expressed, so the structure itself is repetitive, but we can control the sequence. So what we are trying to do with her group is to basically decorate the protein, use this as a kind of molecular wire, and then decorate them with, on one end, again, an antenna mimic, so like harvesting complex mimic, and on the other side with some kind of readout, so for example, either a catalytic side or even just an electrode, to see if we can basically control the transfer of energy from the light harvesting to to something else, basically, and just having a conduit for that.
Nice, thank you.
Any source?
Hello, can you hear me?
Yes.
Perfect. Thank you for a really good talk. I was wondering earlier, you were talking about synthesizing some clusters with these proteins. So I was wondering what types of clusters were you using, and is there an ideal distance or size or material of these clusters that allows the proteins to work as favorably as possible?
Right. So right now we've played mainly with iron sulfur clusters, which are basically very easy peasy complexes. Let's see if I have a good picture of them. Right. And so again, there are compounds that are found in nature, and they're also forming spontaneously in solution. That's what it would look like. We're also interested in using these proteins as nucleating site for example, quantum dots, and interfacing them with materials in that way. We haven't done a ton of work in that area, though other people have And I'll be happy to point in you to some references.
Awesome, thank you.
Other questions, folks? I'll jump in while we're waiting on that. Yeah, and speaking of that, what you just mentioned, Giovanna, that's half the battle, these interfaces. It's just incredibly complex and trying to do this. And I may ask you about that in a minute, maybe when we have more time. But one question in terms of your cobalt hydrogenases, I mean, it's impressive in terms of the oxygenic activity. Did you see a difference in relation to the different mutants you had? Any like was it nominal in terms of the oxygen sensitivity, or pretty similar across? And is that, when you're at that level, you've increased, you're down by a third, but still pretty impressive. What's good enough? Or is it just a matter then in terms of like just trying to increase the catalytic activity of your enzyme?
Right.
You're worrying about that.
Yeah, and so actually, so the main effort now for NTIs, who's a student I have on this project at the moment, is to do directed evolution. So the short answer is no, we did not characterize other mutants, because in the meantime, and I didn't have time to talk about this, but basically we are now using a bacterial display system that we came up with in the lab to make basically libraries in which each bacteria, E. coli cell will have a different mutant basically of the protein. And we thought that would be a much better system to ask those questions, because now we can have a large library generated by either error-prone or designed, and then screen it in different conditions. So rather than doing one by one, that's how we're going to do it. The problem with the library approach is to have a high throughput screen. And so we basically played with several systems used for, one in particular used for hydrogenases. And in our hands, it failed simply because we don't make enough hydrogen. Right? And so the system actually, Kevin Reddin uses this system. It's complicated. We can talk another time, but the bottom line is that we don't make enough hydrogen. So we are now using another system based on a material that basically reacts to hydrogen. And we have some reproducibility issues, but we're working on that. Now, for monitoring reduction of CO2, for example, the easiest thing would be to simply use hemoglobin, right? Because hemoglobin reacts very strongly with CO. And of course, the spectral overlap with cobalt porphyrin is perfect. We won't be able to see much, basically. But that's one of the things we are looking at, basically looking at the increase in absorption when CO is produced. And it's a little tricky, but that's what we are working on. I know, right? So isn't that a shame that they have exactly the same spectrum?
Absolutely. I know, yeah, that makes sense though, in terms of, working on that with the catalytic activity. John, I see your hand up.
Yeah. Hey, Giovanna, thanks for the talk. I wish I understood more of it, to be honest with you, but I think that there was, when we were discussing having these droplets that were catalytically active, it sort of reminded me of some of the work that we were doing in my group, where we actually have these catalytically active colloids. And if the catalytic activity is sort of asymmetrically distributed on the surface of the particles, you can get them to self-propel through fluids. And I was just wondering if you think that there could be anything perhaps similar to that.
Yeah, that would be absolutely, and that will be super fun. Actually, let me stop sharing. Yes, I would love to talk to you more about this, because yes, I think there, a lot of work that has been done with polymers really translates perfectly well to these systems. In fact, the whole theory is basically the Flory theory for polymer aggregation. So I'm sure that, whatever is being done with colloids will actually translate perfectly with this protein-based colloids. Proteins are polymers, right? There's nothing special or magic or whatever about proteins. They're just heteropolymers with the advantage that we can control the sequence at the monomer level. And so I'd love to talk to you more to see what you can do with the systems, because I'm sure we can replicate it and control it with protein-based colloids.
Yeah, that'd be fun. Let's talk about it if we have a
And then
chance.
we can get, we can get bugs to make the proteins, which is even better.
Yeah, get the bugs to do the work for us. Okay,
Absolutely.
thanks.
Definitely, John. We're gonna have Giovanna up once the weather's better, and we'll set something up. Absolutely.
Perfect.
One more question, folks. Anyone? If not, I'm going to jump in with one more in terms of the membraneless carboxysomes. All of that compartmentalization work. I love it. Approaching Rubisco from a different end. Let's not worry about the enzyme. Let's change that environment and up its activity there. I think it's really great. With the when you're talking about light activated activities, and you mentioned this, the problem with turbidity, and I was curious about the trade-off there and what you see in terms of what levels of control do you have over that in controlling the turbidity, and when do you have to change kind of the approach on that side?
Not much, and honestly, so as you probably have observed too, what my group does is very much shaped by the type of students I have, right? So Nick is the kind of kid who has absolutely no problem playing with the instruments and building small instruments. And unfortunately, I'm very happy for him, but he did graduate, a year and a half ago, and that completely changed what we are doing in the group, because in that sense, because it's really an engineering problem, right? It's really not a scientific problem in the molecular sense. It's like, how do I design something that maximizes light absorption in this system? So we really need to come up with a better reactor.
Yeah, and I wonder, because some of the things that we've talked about, if we start looking at multi-compartmentalized systems and things, and we're, at what point do we start increasing it to a level to where we just can't get the light in there? And so we have to think about that from some of these. But yeah, I like handing the problem to the engineers and letting them solve that for us. That works for me. All right, folks. Well, I just want to thank you again, Giovanna, for giving our seminar today, and I look forward to once we're out of permafrost, have you up here for some meetings. I know there's already folks that are looking forward to meeting you.
Same here. Thank you again. This is fun, and again, I'm super bummed, not just for the skiing, but because I was, looking forward to visiting Mira since we first started talking.
Yeah, we'll definitely have you up while it's still ski season, but safe. All right, thanks everybody.
Thank you.
Yeah, and Giovanna, that was great. Really, really, really like that. Took a lot of notes on some things that I think